Our bodies are intricate networks of cells constantly communicating with each other. They do this using chemical messengers that bind to specific proteins on cell surfaces called receptors. Imagine these receptors as locks and the messengers as keys. Drugs mimic these natural messengers. They interact with receptors, acting like keys that fit into these locks, triggering or blocking specific effects within our bodies. This interaction between drugs and receptors is what we call pharmacodynamics. Understanding this interaction is crucial for developing new medications and improving existing ones. Just like a key's shape determines which lock it opens, a drug's chemical structure determines which receptor it binds to and ultimately what effect it has on our body. Some drugs form strong, irreversible bonds with their receptors called covalent bonds. Think of it like gluing a key into a lock. One example is aspirin. Aspirin forms a covalent bond with an enzyme called cyclooxygenase, which is involved in inflammation and pain. This bond permanently blocks COX's activity, providing long-lasting pain relief. Another example is found in chemotherapy. Some anti-cancer drugs form covalent bonds with DNA, effectively killing cancer cells. These strong, long-lasting bonds make these drugs highly effective. However, this strength can also lead to side effects, as they can bind to other molecules in the body, causing unintended consequences. Electrostatic bonds are more common than covalent bonds in drug-receptor interactions. These bonds occur due to the attraction between opposite charges. It's like magnets, where opposite poles attract each other. The strength of these bonds can vary depending on the distance between the charges and the environment surrounding them. Many drugs used to treat high blood pressure, for example, work by forming electrostatic bonds with their receptors. These bonds are weaker than covalent bonds and are often reversible. This reversibility allows for a more dynamic interaction between the drug and the receptor, leading to a more controlled and often safer effect. Section 4. Hydrophobic interactions. Oil and water don't mix, but sometimes drugs do. Hydrophobic interactions occur between water-fearing parts of the drug and the receptor. Imagine trying to mix oil and water. They separate because oil is hydrophobic, meaning it repels water. Similarly, certain parts of drug molecules prefer to interact with non-polar regions of receptors, pushing water molecules away. These interactions are generally weak, but play a significant role in how some drugs bind to their targets. Many drugs that affect the central nervous system, like antidepressants and antipsychotics, rely on hydrophobic interactions. These interactions contribute to the overall stability of the drug-receptor complex, influencing the duration and effectiveness of the drug. Section 5. The right fit. Understanding drug interactions is key. The type and strength of bonds between drugs and receptors determine how effectively a drug works, its duration of action, and its potential side effects. Strong bonds like covalent bonds can lead to long-lasting effects, but they can also increase the risk of toxicity. Weaker bonds like electrostatic and hydrophobic interactions often result in more selective drug action with fewer side effects. Interestingly, even some substances considered inert can have pharmacological effects. For example, certain inert gases can interact with receptors in the brain, producing anesthetic effects. Understanding how drugs interact with our bodies at the molecular level is crucial for developing safer and more effective medications. It allows us to design drugs that precisely target specific receptors, maximizing therapeutic benefits while minimizing unwanted side effects.